All right, thank you guys. Thanks again for joining. So I'll start with introducing Aaron Mack, who is the Senior Engineer 2 at Biogen. So he provides global technical raw material support and expertise in the material sciences group, and that's part of the manufacturing services, right? So he's focused on supplier engagement, raw material innovation, developing robust control and monitoring strategies across modalities, and just ensuring reliability and compliance in biomanufacturing. Aaron has more than 18 years of experience in pharmaceutical research and engineering, including 14 years in cGMP manufacturing. So great experience, and his career spans a lot of different places, including technical leadership and process development, raw material characterization, large-scale manufacturing. He's previously worked also in viral risk mitigation strategies, pasteurization systems, so a lot of great background, and has also implemented an R&D, pilot, and commercial stages. So really has a lot of background in the various types of development as well. Aaron's background from a education standpoint, he has a BS in biomedical engineering and an MS in molecular science and nanotechnology. And he's also a licensed professional engineer. And since joining Biogen in 2017, Aaron's contributed to advancing material science frameworks and enabling innovation, risk mitigation, and supplier partnerships. So thanks, Aaron, for being with us. And then I'd also like to introduce Seth Garrett. So Seth is a senior engineer at Biogen with more than 15 years of experience in the biopharmaceutical industry. He's been at Biogen since 2010, and his background includes over a decade in manufacturing operations, where he's held progressive roles in upstream as well as downstream processing of biologics and oligonucleotides. He has a strong focus on cGMP compliance and process innovation. And during his time in manufacturing, he also contributed heavily to lead of process transfers and materials troubleshooting, and supported implementation of advanced analytics as well, such as HPLC at the manufacturing level. So his current role, Seth is a member of the material sciences team, also under the manufacturing sciences organization at Biogen, and specializes in single-use technologies and technical support across all the global network that Biogen has. He also is a technical SME, and this includes areas like supplier capability assessment, regulatory support, executable and teachable studies, supply chain continuity, and he also has additional job duties on top of everything else, including the standardization of single-use systems and development of raw material strategies and authoring of regulatory submissions. So really passionate about continuous improvement, and he's helped lead cross-functional initiatives to align Biogen's global material programs with the evolving regulatory expectations as well. Seth has a BS in biological engineering from North Carolina State University. So welcome, Seth, Aaron. Thank you so much for being here, and hand it over to you guys.
All right. Wow, rousing introduction. Thank you so much.
Yeah, for real.
Yeah.
So our discussion today, we really wanted to come and share with you guys what our experience has been dealing with the pre-made solutions that we are getting from Thermo and using in our manufacturing space at Biogen. So really, from the introduction side, again, Aaron, I'm Seth. I would say probably the simplest, easiest way to just sort of explain the material science group at Biogen. We are an arm of our company's manufacturing sciences, or MSAT, organization. And within that, I would say probably the simplest, dirtiest way to explain what we do is that we deal with everything and anything related to materials as it comes into the company or as it's going through the company and potentially out the door. So what we wanted to talk specifically about today, discussing the implementation of pre-made solutions, we really wanted to talk about what kind of led us to the idea of thinking, hey, why don't we consider outsourcing buffers? We also will talk then about the ID strategy of how that works now that we're getting these pre-made buffers, especially as we start getting a whole bunch of them and thinking about the fact they're essentially all clear liquids. How do we differentiate between all these different clear liquids that are coming in the door to know what is what? So looking at that, and then also from an impact standpoint, how has that worked out for us? You know, what have we gained? What have we lost? Just walking through that entire process. So, just a couple basic disclaimers. Aaron and myself are here as representatives of Biogen, but that being said, our views and opinions are really our own and are not reflective as the general overarching feedback of the entity that is Biogen. And then similarly, we have a thermo disclaimer as well. You know, we're not necessarily trying to specifically endorse or recommend or promote any specific thermo scientific products. So just to have those two pieces out there. But let's get past that and just get into a little bit of the basics of Biogen as a whole. So the company itself was founded actually in 1978. Most people probably wouldn't know, and that's understandable. But if you were to say, go to our 5000 RTP site that I know Trent and Teresa at the very least have had the pleasure of visiting on multiple occasions, we have street names there such as Phil Sharp Drive or Wally Gilbert Drive. These are some of the founders of our company. So they are some of the folks that got together and kind of came up with the idea of the company that eventually would become Biogen. We are headquartered then in Cambridge, Massachusetts, but with that being said, we do have global manufacturing capabilities both with our RTP site in North Carolina. So let's see if I can get this to work. That is not really going to work well. That's what I figured. Okay, great. So our RTP North Carolina site, which technically there are two sites. We have a 5000 Davis Drive location and a 900. They're right down the road from each other. And then we also have a manufacturing facility over in Solothurn, Switzerland as well. So between those sites, we ultimately have 7 different manufacturing facilities available across a whole selection of modalities. So we deal with a lot of biologics, a lot of ASOs. We had dip our toes a little bit into gene therapy, maybe not so much these days, but we do also look at parental fill as well. We're starting to look into label and pack as well. So a lot of different areas for us. And if you were to sit down and look at all of our factories together and all of the various capabilities between all of the different tanks and bioreactors that we have, we're talking about roughly 263,000 liters of installed bioreactor capacity. Shout out to Solothurn. I know they've definitely got a lot of it with that facility. But even outside of all that, so our manufacturing network does also include a lot of external manufacturing opportunities. We definitely partner with a lot of different groups. Definitely thinking about Thermo at the very least, the old Patheon Ferentino site. I know we definitely have some dealings there. So really just trying to do whatever we can to help ensure resilience of our supply chain as we're going out here and producing all of these various products. So talking then about premix solutions and how we got here. So the problem arose, I want to say, probably about a decade ago at this point. We had, in one of our facilities, had the very fortunate luck of gaining an extra production bioreactor, which that's fantastic because that allows us to now create yet another round of products. Really, a lot of our process is going to be driven by our production bioreactor. So as we have originally only 3, now we've added a 4th one. That's fantastic. We can potentially have a lot more throughput in that facility, but there's a bit of an issue with that, and that if we're now going to potentially increase throughput of our facility, how are we gonna manage that? Because we're not looking at necessarily hiring more people. We're gonna try to keep the production schedule at the rate that it's been going, just now with a 4th production bioreactor. And if we can't do any of that, and we really need to focus all of our people more on, say, purification activities as we hit downstream, we need to figure out some way to work smarter and not harder here. So that brings us with the idea of, well, why don't we look at trying to outsource maybe some of our solution prep work? We have the capacity for it, but we probably aren't going to have enough to continue to support more batches on top of what we're already dealing with on the regular. So considering that, and then considering the fact that in reality it could take easily 10 to 12 hours, possibly more, of time to take 2 MAs basically away from the process to go do solution prep work now, is there a way that maybe we can pull them back so we can get them back to doing work in purification instead and not having to just sit down and do solution prep? Combined with that, then also there's the fact that some of these cases, some of these solutions that we're making are ones that we have to make multiple lots of per batch, because maybe it's like a sodium hydroxide where we need to chemically sanitize something, or I don't know, some sort of sodium chloride perhaps. Just something where we know we're going to have to prep a whole bunch of lots of it because our tanks are only so big, there's only so much we can do per prep. Just are there ways that we can sort of speed this up? And so, yes, that led us then to the idea of, okay, well, why don't we talk about outsourcing our solution prep work, work with some trusted suppliers that we have then, and figure out if there's some way that we can sort of make that work, looking at, trying to create some sort of economy of scale based off of that, and, are there potential savings that we can have in the long run, not just in terms of man hours, but also potentially just in terms of raw materials. So, yeah, as a result then, we decided the smart thing to do, let's go ahead and outsource our solutions. That way we don't have to add more equipment, we don't have to add more FTEs, we can keep everything else consistent, but still be able to increase throughput. And Aaron, I'm going to hand you this clicker.
All right.
And
so
let you have at it.
as far as those challenges go, with externally prepared solutions, we found that we need to have at least 6 months of stability. That's the bare minimum, because it takes a month to receive and release material, can take a little longer sometimes. And then to get it into manufacturing, there's usually a couple more months before we actually get it onto the floor and used. And that's in the best case scenario. A lot of times schedule changes can throw us off. So we determined that at least 6 months of shelf life are required compared to days for in-house prep. And I think for in-house preps, we have up to 60 days to use those as well, because usually, we're making them and then we're using them pretty immediately, and we didn't have a lot of direct stability work beyond those 60 days. So our group participated a lot in effectively setting up the stability work, and also leveraging a lot of what our suppliers have already done. Thanks for the sodium hydroxide stability work, appreciate y'all there. And then additionally, there was sometimes a lead time of like 6 to 9 months to establish a new item and procure this item from suppliers. So from the day we say, hey, Thermo, we need this material, they set up a custom, what we call it, CRD, custom requirements document, which is effectively a user requirement spec for these individual products that you guys are making for us. And then from that point, we can expect it's going to take about 6 months before the first order comes in the door. Maybe a little bit longer for things like custom media, but that's a different story. So large batches of pre-made solutions do require substantial warehouse space. We can put 4 by 200 liters onto a pallet, and well, in the EU only 2 by 200 liters because of the smaller Euro pallets that are required there. And then QC testing is required to confirm identity and correct formulation. So appearance and identity are 2 main things that I think are always required on anything that comes through the door. Therefore, we came up with this solution identification strategy. So as I said, ICH-Q7, we need to be able to receive and release these based on appearance and identify what they are. Seth mentioned earlier, there are a bunch of clear solutions, so appearance is going to be, okay, solution's clear. What else do we got? So our strategy is to use at least 2 tests, mainly pH and refractive index, to identify these materials. And then you'll see there's a few groupings here where there's kind of a large number of overlaid materials, so to speak. And that's where our third test of conductivity comes in, where we can differentiate further if there's too much overlap between pH and refractive index for 2 different formulations. There is one formulation that does come to mind that doesn't fit in here that we do have to use ICP-MS for, just because of the trace amount of manganese that's in one versus another. But beyond that, though, we do have 24 of these pre-made solutions uniquely identified. And if you've ever done pH and RI, these are very quick tests. And our QC lab loves that. We don't have to sit there and wait for an HPLC or ICP-MS or other things like that. And then conductivity as well are fairly quick tests overall. And some folks will say, well, why don't you just use pH and conductivity? Well, conductivity isn't quite as robust as refractive index. When it comes down to it, there's things like heaps buffer that have an inverse conductivity, but kind of throw you for a loop sometimes. But that said, in general, as long as we have pH and refractive index and then conductivity as a tiebreaker, it's proven to be robust, and our regulators really are happy with it at the end of the day. All right, so once again, pre-made solution stability. So all of our solutions had to have at least 6 months of stability. I think the shortest stability time that we had to this point was about 1 year. I think that's with the 1 molar tris, if I remember correctly. But overall, we were able to leverage some of our supplier stability work, 24 months for acetic acid and sodium hydroxide solutions, with the caveat being that 0.1 molar sodium hydroxide, I think that's around a year as well, just because of ingress of CO2. We'll throw the pH off on that material at about, I think, 15 months or so. But yeah, as you can see here, though, they're very well differentiated in terms of pH. We have osmolality as another, let's say, counterpoint to conductivity. They're not exactly the same, however, they both depend on the polarity of the solution. And then refractive index, where a lot of times we're only doing down to like 4 decimal places, but we do test out to 5 decimal places. So yeah, these don't seem so different. However, they're different enough such that once you get down to the 5th decimal place, very easy to effectively separate. All right, and then stability for all others, takes about 1 month. 1 month in for data complete of the trend, 3 months before the orders are placed. So right around the 3 month point, you're like, does this look like it's going to be stable or not? And then we can tell our procurement folks, yeah, it's good to go. We'll have the report done by the time the material arrives. So a lot of times we're going by the skin of our teeth with some of these new pre-made solutions overall. Would you like to take this one?
Sure.
Yeah. So that's my bad guy. Yeah.
Yeah, so really the biggest thing was trying to land on a design. I will say in the very, very distant past, we looked at potentially 500-liter bag design, but that 500-liter bag size tended to get a little bit tricky because we then have to manage totes and shipping them back and forth, and I think that turns into a headache for all companies involved. So really following on a 200-liter bag design really turned into being really the path forward. So we came up with a pretty standard agreed upon 200-liter bag design. It's pretty straightforward. You do have a fill line going in, so you could connect it up to whatever your buffer fill manifold is as you are producing it. You can fill the bag, and then we'll have line number 3 here with the AQG connector on it. For Biogen, that is really kind of more our preferred at this point, as much as we can, is to start moving towards the aseptic quick G connector. So we have something that can easily then connect up to any number of other parts or systems that we have, whether it's, we're attaching just a simple Y onto it or attaching up to some kind of larger manifold to make multiple connections of multiple bags. This gives us all those flexibility, all those options. Beyond that, it's very important for us to make sure that we know what the bag film is and what the materials are, because leachables and extractables are always a very big concern for us. That is absolutely something we have to scrutinize with any of our products. There's always assessments we're looking at doing. Aegis 514 is really our preferred where we can, definitely for any potential concerns as it relates to, say, ergophos. Just making sure that we can have something that is ergophos free really is preferred. Polypropylene fittings, just going to be in general, good idea for pretty much any solution that we have. We'll use silicone gaskets for pretty much everything across the board. PVC tubing, we have definitely used a fair amount of that in the past. I know we've started to move away from that a little bit. I know there's a few folks here that I've had to reach out to in the past about making some changes away from it. But and then beyond that, C-flex tubing if there's other spots that we need welding. Braided silicone tubing if we're going to be potentially dealing with like a higher pressure, higher flow rate kind of application, so that we're not either crushing or potentially exploding tubing. And then we've already talked about the AQG. So
I will say, it does say bottom drain bag here. That's really not a requirement overall. I know our operators tend to prefer a bottom drain bag because air bubbles are a thing. We use diaphragm pumps, and priming those diaphragm pumps can be a little bit troublesome.
yes.
But as long as you have some kind of bubble trap or something like that in line to be able to pull that air out of the way effectively. I don't know if you guys have ever drank Capri Sun. But you can, the way you drain that bag is similarly. It collapses as long as there's not too much air in the way, and as long as we have 6 feet of tubing so that the tubing can get onto the ground and connect, and we're not having things like kinks form. That's another like watch out too. We in general have had great results with these types of bag designs.
Yep.
All right, what's next? All right. This is a good one, I think, overall, in terms of site impact. So with our legacy method, we had to make 23 internal batch preps for, let's say, a single process overall. So that's what was like 2 weeks of your time when you were an operator back in like FVM and 2K. And really, this is not very stimulating work for operators, you know, turning and burning salts into buffers that are going to be used, and then sterile filtering them into bags, and then sending those over to the warehouse to be stored until they're needed in production. I guess it could be a little bit more mind-numbing, but notice though that we've gotten down to 1.25 external preps per batch. So since you guys are able to make so much bigger batches, what was it, like 9,600 liters, and provide those to us, that basically disperses all those preps. It makes it a lot more efficient overall. So our internal buffer prep FTE hours went from 250 to 0. Nice goose egg there. Everybody loves that. And whereas though the materials cost per batch, it increased a little bit, but time is money at the end of the day. And it's a lot, actually, this costs a lot more than this at the end of the day, especially when we can have our FTEs focused on things that are more value add, whether, effectively attaching these buffers to our chromatography skid and getting our product off of that and down the line to UFDF. Right.
So, in conclusion, we ultimately implemented this premix solution setup and didn't find that we had any changes to our solution release criteria. It only gets tested by our suppliers, and then ultimately does get verified still at Biogen. So our QC folks are still getting ultimately what they need. We can still test it and confirm that it is the material that we all say it is. Stability data, we were able to collect that over time to be able to support any extended solution expiries that we have or potentially find ourselves requesting in the future. We ultimately will have these solutions filtered with the same sort of filters that we would be using at Biogen anyway, so we're really same there. The bags are still going to be comparable to the bags that we would have been using on the floor in manufacturing anyway, so it would have still been some sort of Aegis bag film. Solution comparability, we were able to confirm that in line during our chromatography process. We do have inline sensors. We could also take offline readings if for some reason we felt the need to do so. And at the end of the day, like even in say a worst case scenario where for some reason we can't get enough of a premix solution in for whatever batches that we're making, that's okay. We do still have the legacy solution lot records available that are still potentially able to be put together and mixed in the event of some kind of supply disruption. Obviously that's not ideal, but we do still have backup plans essentially. So we haven't necessarily put all of our eggs in that basket in the event of some sort of disruptions, but that's really all we've got.
Yeah, we're going to thank these team members. Dave Kolwick, who was the old leader of material science when we started doing this work. He's the reason I have a job over at Biogen, so good on him. Yesenia did a lot of our own internal stability work. Gitanjali did a lot of the testing and effectively determining refractive index was the best path forward. We still have the little K patents RI that we effectively use in manufacturing. I still use that on a regular basis down in our lab. And Andrew Hedgepeth, he was actually integral in implementing this in our 2K and FEM facilities. Gave us a lot of feedback on those 500-liter totes too. Don't do this kind of thing. Becca Hawthorne for QC lab representation. Patrick Taylor for a lot of the vendor, what would they call it? I forget what they're called now, like GRV, global, they're effectively our QC folks. Pete Murphy, who was integral in setting up new items for us as well. Rick Cross, Holla, our old friend there. And then thank you all as well for helping us set these things up and implement. I know it wasn't easy at times. There's a few headaches that we had to work through, but you enabled success. And I think we're able to make 30% more batches out of this facility because of this project.
Yes.
That's huge, when we're going from 60 to 90 lots of drug product, or drug substance, I should say, that are coming out. So it really enabled us. It really enabled our partners that we use a lot of these materials on as well. So you're making us look great. Really appreciate that.
So, thank you.
Really, in-depth presentation. We did have a couple of questions for you guys as well, based on some of the information that you shared with us. Aaron, maybe to start off with you, you talked about some of the challenges initially. Could you maybe get into a little bit of details on how you align some internal stakeholders? Because I'm sure this was not the most easy conversation to have about outsourcing buffers.
So anytime you talk about this kind of outsourcing stuff, a lot of the operators are like, well, am I going to have a job? So we never framed this as removing FTE hours. We always said it's repurposing FTE hours. There's a lot of important things that we need to do, and there's a lot of other things that are, while they're important, they're much less important when it comes down to being able to enable drug substance release at the end of the day. So that was something that I think was drilled into my head by my boss. Like, no, we're not getting rid of any FTE hours. We're just shifting them to a different area. Additionally, our QC folks, they like to test stuff, quite frankly. So if we're not doing all this salt testing, what are we going to do? We've still got plenty of other testing to do, friend.
Right.
At the end of the day, it's just going to enable them to be able to focus on the tests that they need to do and more quickly be able to turn those around. So by moving from, let's say, all the compendial testing that we would have to do for like all the salts, and some of those are outsourced tests and things of that nature, there's a cost to that. We are able to just do pH, refractive index, and appearance. And that makes life a whole lot easier for the guy that's doing the receipt and release testing overall. And then they can get on to the other multi-compendial testing that we have to do for things like excipients that we're using. So that's the other part. None of these materials are used as excipients. That's kind of where we drew the line, just based on risk to the patient as well. So everything is used effectively upstream of our UFDF process, which that inherently has excipients in it. So with that framing too, we did have to include all those folks that were a part of a cross-functional team and get their buy-in at the end of the day. And then I think once a couple months worth of meetings we got through, everybody was fully on board. And since we've implemented it, I haven't heard complaints about it. They're like, we're getting more of these pre-made buffers, right? Yeah.
Sounds good. Thanks, Aaron. That's super helpful. And then Seth, I guess one of the things that you had mentioned was also risk mitigation strategies, right? So could you get into that a little bit just from a supply chain side, and have outsourcing buffers helped with some of that? And what are some of your strategies now based on outsourcing versus having in-sourced before?
Yeah, I mean, I think from really a risk mitigation standpoint, again, we've always got the fallback option of if we needed to mix solutions, we can do that.
Sure.
It's not the end of the world, right? I mean, those are all raw materials I would say that we already would have in-house for other preps. There are a lot of cases where we may be doing smaller preps or more unique buffers that ultimately we would have needed to make anyway. So it's not really a big issue, I think, for us to still have those extra materials on hand. We're not potentially putting anything to waste because of it. So I think that very much helps us across the board in terms of just making sure that we have what we need at the end of the day to be able to make these batches and push them out the door.
Okay, yeah, that makes sense. Thanks, Seth.
Yeah.
I think, Aaron, back to you then. Question around just the initial stage of thinking about outsourcing. Anything you would have done differently given what you know now?
Well, quite a few things now. At the time, we only really had the capability of doing buffer dilution. So we can, I think we do somewhere between 5x and 15x dilution of what is effectively a complete buffer. One of the concerns from our TD group is like, well, what's the pH on that stuff? The pH is really important. Yeah. pH gets tested when it goes through, or right before our chromatography skids as well. So that was one of the things that mitigated their setup. But that said, I think the future holds buffer conditioning skids instead of buffer dilution skids, wherein we have, let's say, between 4 and 7 highly concentrated components that are all going into this skid that has a static mixer and a connection to our Wi-Fi port, such that we can make complete buffers on the fly. As long as we have those pre-dissolved solutions, we're not worried about salts in there or anything like that. The idea would be we'd have a break tank that would effectively be ready to collect a buffer that comes off of that skid, and then we could make any number of different buffers for our multitude of different processes. At any given time, we have something like 15 programs running, from things like ASO purification, which oligosynth nucleotides. They don't use any cells or anything like that. We're synthetically making peptides, which is where our product Spinraza it comes from, effectively, versus our large molecule processes where we have our own Biogen programs, as well as, last count, I think we have 5 different partners that we effectively help make their large molecules. So they all have different unique processes. However, all those different buffers are made of mostly the same components.
Right.
Sodium phosphate, dibasic, monobasic, tris HCl. The list is surprisingly short, quite frankly. So being able to make this type of change to a conditioning skid, I think, is going to be really enabling and allow us to maybe eliminate a few more buffer tanks and get another bioreactor, and so we can make a few more batches per year.
Sure, sounds good. And then specifically, I guess, that process of insourcing to outsourcing, any specific things in that process itself that you would have done differently now, given what you know?
In terms of, I guess, making it easier to do overall, we're not the best at being able to turn some of these things around sometimes, because, again, we have a lot of partners and they all have to have a say in a lot of these things too. So, and then being able to more easily show those partners the benefit of doing this kind of outsourcing work. That, I think, to me, might make it a little bit, I think we've got a pretty good standard way of doing this now. However, it still takes like 6 months to get a new product in, so anything we could do to move that a little bit more quickly would be great.
We have robust quality systems, but they're not built for speed.
That's true.
And anytime we do have to consider any sort of partner approvals or acceptance of it, that takes its own amount of time. So some of these changes go slower than we prefer them to.
Sure.
Absolutely.
That makes sense. I guess that's kind of the flip side of that question. Anything that surprised you, or any unexpected benefits, kind of given the implementation of the outsourcing strategy for buffers?
Really, benefits were the only thing that I ever truly remember of it. Just thinking about, definitely looking